Okay, everybody, we're going to get started. I want to welcome you all. I think you know both Richie and I, but if you don't, I'm Ned and
I'm Richie.
he's Richie. So you can tell that we've been a couple for a long time and continue together. Believe it or not, this is our 29th meeting, consecutive meeting, with the exception of the COVID year where we had to take off from that. Next year is going to be the 30th, and I want to acknowledge Amber. Where's Amber? Amber's over here. Amber, stand up for a second. Amber's our coordinator who's done an absolutely terrific job organizing and keeping Richie and I on task, which isn't easy, as you guys can imagine, and also has told us that she's thinking of something special for the 30th, which is next year, so we're excited about that. This is a very special meeting, we think. It's focused obviously on emotion and affective neuroscience and psychopathology. That's been a theme over the years. And one of the really special things about this meeting, which Richie will tell you more about, is the time that we've devoted to the speakers to speak, the quality of our speakers from the standpoint of the body of work that they've done, and also the time for discussion, which is 45 minutes. And Richie will tell you more about how the speakers are going to get grilled, so you guys can start to have a little anticipatory anxiety about that. So I'll just say a few words about the lineup. You've all seen this, but again, we're going to start off with Kerry Ressler, and Kerry's going to talk about PTSD and his translational work, which has been amazing. And then Olu Ajalor is going to talk to us about his work with connectivity at a variety of different levels, which is going to be exciting. Alvaro Pasco-Leon will talk to us about TMS, history of TMS, and latest developments, which we're really excited to hear about. Raquel Gore, who'll be coming in later, has done amazing work at University of Pennsylvania related to neurodevelopment, some focus on schizophrenia, but other disorders. And Tor Wager is going to talk to us about his work that's been broad and related to empathy and a variety of other exciting topics related to emotion. So it's a great lineup. We've encouraged the students to really let their guards down a little bit and to not be afraid to engage with the speakers. And we've encouraged the speakers to do the same. So we really want to see this as being quite interactive. One of the really special parts about this meeting has been our ability to have travel awardees. This is really gauged towards trainees in a sense, the whole meeting, but we've been able with outside funds and support to be able to provide funds for folks to travel here. And we believe we have 11 or 12 trainees today. You guys want to stick up your hands just real quickly, just to show who you are. And these are students and folks from all over the country who are doing really exciting work, who we've been able to select to come here and will be part of the meeting and also part of the poster session. So we're really excited about that. And then also there's a poster session. It's at 4:30 to 6:30 with food and drink, another opportunity to interact, and would encourage you all to hang around after the talks to engage in that as well. Let's see. This is just a reminder that tomorrow we also have breakout sessions, and this is an opportunity for everybody to get to know the speakers a little bit better, to circulate around and so on. So Richie, do you want to say a few more words about the meeting, and then we'll come back and say a few more remarks?
So it's great to see all of you, and Ned and I are still at this, and it's still really a lot of fun, and the work that we're fortunate enough to bring to all of you continues to be super exciting, and we have a great lineup this year. As Ned said, this is an unusual format. It's really going back to how science was once presented, but rarely these days. So every speaker has a full hour to present his work, and a full 45 minutes for discussion. And the trainees from Wisconsin will be leading off each discussion period. And one of the honors that I get is each spring semester to convene a seminar of the trainees at Wisconsin who are part of our T32 that is focused on emotion research. It's a training grant from NIMH that we've had for more than 30 years now, continuously, that funds both pre- and postdoctoral scientists who are working in this area. And the reading list for that seminar are the papers that the speakers nominate as their 5 papers that they want all of us to read. And so I can guarantee you that those papers were read carefully. And that's what the seminar consists of. And so the trainees will lead off the discussion after each speaker. So it should be really exciting. And I want to just also encourage all of you to have a very low threshold for jumping in, including co-speakers. And this is often the most lively part of the symposium. So we're really looking forward to it, and thank all of you for being here and contributing.
Great. You can see the different rooms that everybody's in. I just noticed that I'm in the Mesozoic garden. I'm not sure why Amber put me there, but there must be some intent there. We also have a virtual audience, and I'm not sure how many folks we have on virtually, but about 40 folks. And so we'll be having questions from them too. And during the discussion section, we'll try to alternate as best we can across that. I want to really acknowledge and thank our supporters. And, we've been very fortunate to have the support of the Department of Psychiatry, the Health Emotions Research Institute, and the Center for Healthy Minds as all part of entities here at Wisconsin that both Richie and I have been very, very involved with over the years. But also importantly, Corcept Therapeutics has been a partner of ours for at least 10 or 15 years now, and has supplied us dollars that have really enabled us to continue with our travel awardee funds. And that's been a really, really important thing. So I want to acknowledge the support of all these entities, and especially Corcept Therapeutics. So with that in mind, we're ready to get started. And we're going to start off with Kerry Ressler, and Richie's going to introduce Kerry. I should just mention, I think this is your second time with us, Kerry. A decade ago? Wow. Okay. So Richie, go ahead.
Yeah, there have been a few very special speakers that we have had on more than one occasion, and it's really an honor for me to introduce Kerry. Kerry is one of the leading experts in the world on the effects of trauma on the brain. Kerry has pursued an extraordinary program of both basic and clinical translational research on this topic. He has been interested in a wide variety of issues that all come together to inform the many different influences that we need to account for in order to understand how trauma affects the brain and produces psychopathology. I've had the pleasure of getting to know Kerry in a few interesting different contexts over the last few years. And one of the things that is also really apparent is that he's not only a great scientist, but he also deeply cares about human suffering and its alleviation, and is really dedicating his life to the pursuit of science in the service of that. Kerry has many honors, which I'm not going to go through, but we're really so fortunate to have him here today. So with that, I'll have him come up. Thank you.
Thank you so much. Good to see you. Thanks everybody for being here bright and early. Can you hear me okay and lavalier and everything? Great. It's a lot of folks who come, obviously the people who come to this are speaking all the time in lots of places, but there's something particularly special about being able to speak at the Ned and Richie show on emotion. So this really has a reputation and is just a wonderful place. And I hope you enjoy, I think I will enjoy it as much or more than you do on the course of the discussions over the course of the day and tomorrow. So I am going to give sort of a smorgasbord of our work and the field's work over the last 20 years on post-traumatic stress disorder, trauma, emotion, really focusing on negative emotion and how we study that across species. I think I'll just jump in. So to start, Okay, so just put everybody on the same page about when I say post-traumatic stress disorder, what do I mean? So PTSD is defined as having a severe life-threatening trauma in which one has ongoing symptoms, at least that are at least a month after the trauma. So the first few weeks, I'll talk about some of our emergency department work for the development of trauma-related disorders. The first few weeks we call acute stress disorder because most people recover after a trauma. The majority of people, about 90% of people after any one trauma, will recover to pretty normal premorbid symptoms and functioning before the trauma. But about 10% will go on to have significant symptoms. 1 month after the trauma, we call it post-traumatic stress disorder. 3 months after, we call it chronic PTSD. The symptoms that occur are 4 clusters. The first is really the most classic for PTSD. It's these intrusive flashbacks and nightmares, essentially reliving the trauma memories. And as I talk about PTSD as a disorder of memory, we can really think about, and now we understand things like engrams, that these engrams we think are being replayed really in vivo and of their own volition. And people with PTSD feel like they're being haunted by these memories. And they can really be, extremely dysfunctional, both nightmares to the point of just not sleeping and hyperarousal every night, and all the chronic symptoms that happen with that, as well as flashbacks that can occur anytime. People who may have been attacked, maybe having an interpersonal conversation and then, get triggered and are in a totally different space. The next set of symptoms is avoidance, and that's really sort of avoidance of people, places, and things that trigger the trauma. And that is, of course, a natural thing to do if one is afraid of dying or being attacked. Within that space is also dissociation that we don't explicitly talk about in DSM-5, but we know happens very much. And I'm not going to talk about it much today, but with people with a subset of people with PTSD with very severe trauma will dissociate to where they're feeling separate from their body, separate from their mind, don't feel like they're present. This often happens with people with childhood trauma. And as you hear more about through the course of the day, precision medicine, with PTSD as with the other disorders that we'll be talking about, there are likely dozens, if not 100 different types of PTSD. And one of the first layers of cut is more the dissociative type versus the non-dissociative type. And when we talk about hyperarousal of amygdala symptoms and threat hyperarousal, you see that very clearly in the non-dissociation. But with those dissociation, you see this very shut down type. Along the same line of being shut down, negative alterations in mood and cognition. People experience very severe depressive symptoms, and it can often be hard to separate classic depression from trauma-related PTSD and depression. And then poor concentration. When your mind and brain are focused on survival, it's very hard to have executive functioning. And then finally, the other component that is most specific to PTSD is this alterations in arousal and activity. Startling easily, I'll show you some data from startle across species, feeling jumpy all the time, not being able to sleep. And while it's not explicitly stated in DSM, one of the most critical things that people will say is, I'm always afraid and I can never be safe. And as we think about translation to this concept of threat and fear, this holds very strongly. So as a psychiatrist, the components, it's a very disappointing area because really what I'm going to be starting with today is the disconnect between where we currently are in our treatment availability on the ground versus where the neurobiology and genetics are. There's a huge amount of promise, but we haven't yet reached that promise, which is both exciting and somewhat disappointing. And we hope that your generation can make it further than we have so far. In terms of the current, this is my only treatment slide, just to sort of catch you up with the very limited treatments we have. Actually, from a therapy perspective, we have quite a few treatments. And really the first line for trauma-related disorders is exposure-based therapy, the various therapies, cognitive behavior therapies, with prolonged exposure and exposure-based therapies, the ones having the most data for them. There's a number of subtypes of this that go along with having sort of emotion regulation strategies, mindfulness strategies, dialectical behavior therapy prior to exposure. Other things that are essentially helping the person handle their emotion and that hyperarousal before they start doing the hard work of exposure in many ways so that they don't either dissociate or avoid and leave the room and not come back to therapy. What happens with exposure therapy basically, and I'll start and end with this figure with some animal models at the end, is this concept of extinction. And a therapist might use a term called SUDS, or subjective units of discomfort, and say, When you feel so anxious, you're going to have a panic attack or feel like you're going to die, that's 100. And when you're so bored, you're going to fall asleep, that's a 0. So at any given point, where are you on this 0 to 100 scale? And the first time the patient comes in and is talking about their trauma, they're going to go close to 100. And the job of a good therapist is to try to hold them there and not go too high, but not let them avoid completely either. And over the course of the session, they'll actually calm down and it'll be sort of okay. And they'll keep doing that over and over again. And over the next session and the next session. And eventually this trauma that it leads to the triggers and leads to dysfunction has just become a bad memory. And so a lot of what we talk about when we talk about Pavlovian extinction of fear is essentially relearning or retraining the brain about safety in the presence of trauma memories. In terms of medications, we actually only have 2 FDA appropriate approved therapies. And that's sertraline and paroxetine, 2 SSRIs. They're essentially treating non-specific symptoms as we treat anxiety and depression with these drugs all the time. In the discussion, I can talk more about how some of the other off-labels may work and may or may not work. One of the most interesting is prazosin. That's probably the only thing in our armamentarium that really fits within our understanding of the neurobiology, in which you have this very clear hyperarousal, hyperactivated sympathetic state, and blocking alpha-1 adrenergic receptors can be very helpful for hyperarousal and nightmares, but the dosing and the implementation of that is quite complex. So, to start off, I just want to say there are no current FDA-approved drugs based on our understanding of the neurobiology and genetics, and yet the field has made tremendous progress in the last 20 years. So our goal then is to take over 100-year-old data and understanding of behavior and threat memories from Pavlov and translate that to PTSD. And there are several components that we feel like lead this to be quite tractable. One is that I'll tell a lot about the basic neurocircuitry of fear. Today, I'll, in addition to doing a little bit of work that I'm sure you've heard about before, amygdala work, I'm going to tell you about some of our sensory system work that we think is quite exciting too. Second, we know when it starts. By definition, PTSD starts at the time of the trauma. That's one of the few places in psychiatry where we can say that, and therefore we can learn a lot about what's happening in that early aftermath of trauma, what's happening longitudinally after trauma, and we can model it. And I'll talk a lot about consolidation of memory as these fear memories become overly consolidated or overly instantiated. And then finally, thinking about PTSD as a learning and memory process. Often we think about neuropsychiatric disorders of memory as dementia and loss of memory, whereas PTSD we can really think about as an overlearning of this initial threat memory and then an inability to learn new safety memories. And we can try to translate some of the great progress in synaptic plasticity, learning, and memory to these models. And the goal of all of this is to have a predictive biomarkers of both treatment, response, intervention, and brain functioning for novel interventions. So starting with Joe Ledoux's classic picture from, I guess, 30 years ago now. So my overused story, but I still find it fun, is when Betsy and I, my wife and I lived in Atlanta, we would go walking through the woods and we'd be hearing the birds sing and we would be having a nice conversation. And then, oh my God, Betsy's 10 feet behind me, jumping up and down. Oh, I'm like, what's going on, Betsy? She said, it's a snake. It's not a snake, it's a stick. It's always a stick. But Betsy's a snake phobic and her eyes and her brain are ready for the snakes of the world to get her, right? And so we can all have stories like this. We all have little phobias that, are fine. Or you turn the corner and there's somebody there and you jump. So in normal day-to-day activity, we can all experience a situation where we've activated our fight or flight reflex, if you will, but mostly we can calm it down very quickly. So what's going on? So all of our sensory systems, and I'll dive a lot more into the visual system later, are attuned to the world around us all the time. And they're receiving immediate information from the world, and they're receiving this non-processed stick or snake-like figure image, which is going to in a couple synapses to the visual thalamus, and then in one more synapse to the amygdala. And then in parallel, that information is going to primary visual cortex, secondary upper level visual cortex, parietal cortex, prefrontal cortex. So it takes about 100 milliseconds for the amygdala to receive this preprocessed information and about half a second for the rest of brain functioning and what we probably think of as consciousness to receive this information. The amygdala has already set off the hardwired fight or flight reflex long before you're consciously aware of what's going on. You've already jumping, your cortisol, your adrenaline, everything we'll be talking about is going, and then, oh wait, it's just a stick. If you're well modulated and you don't have PTSD, your heart rate's still going to go for a little bit, you're sweating a little bit, but you're going to be okay. If you have a trauma-related disorder, or many of the anxiety disorders we'll talk about, it can take hours to calm down, or you may go from one trigger to the next. And so in many ways, this system that is evolved and really conserved from all the way from the lowest vertebrate or mammals and even reptiles to humans in terms of amygdala function probably causes more harm than good in the modern world. So I won't talk a whole lot more about this kind of amygdala circuit, but just the take-home messages here is the amygdala is constantly receiving information from all the sensory systems. It's processing that in a very rapid way with auditory and visual and other sensory cues coming from the lateral and basolateral amygdala, that information going to the central, and the central going to multiple brainstem regions. Work in the 70s, 80s, and 90s worked out the hardwired connections from the central amygdala to multiple subcortical and brainstem regions that lead to all the symptoms of a panic attack. That symptom of increased heart rate, blood pressure, GI distress, respiratory distress, arousal, increased startle, freezing, decreased social interaction. PAGs also fight or flight, and sort of some of the aggression responses I'll talk about later. And then the paraventricular nucleus of the hypothalamus, which leads to the corticotropin releasing hormone, HPA axis system that we'll talk quite a bit about. So speaking of HPA axis, I'm warm and going to take my jacket off. I'm from the South, and I used to be warm, and now that I've lived in the North for a long time, if it's hotter than about 60 degrees, I'm sweating. So okay. So this is one of the few places in our field where we really have a hardwired reflexive circuitry that explains a whole set of symptoms related to a symptomatology. And we may call this a panic attack, but it's the same thing you see in a flashback in a triggered threat response. The amygdala is constantly being modulated by prefrontal cortex and hippocampus that we'll get into more. And before I dive further into the research we'll talk about today, I also wanted to introduce the Grady Trauma Project. So in addition to PTSD being a disorder of veterans, and this is, in its current nomenclature, was really started talking about it in the 70s after the Vietnam War. And then shortly thereafter that, with the women's movement and work by a number of folks looking at at sexual assaults in women, and the women's rights movements really led to the increased appreciation in the 70s of trauma in women as PTSD, but not really until the 90s with work by people like Naomi Breslau and others realized that trauma in our inner cities and in our under-resourced communities is bad or worse than many of the war zones. And so we started in 1999, 2000, the Grady Trauma Project at Grady Hospital in Atlanta. Since then, we've interviewed about 13,000 people. This is now led with initially by myself and Beck Bradley and Tanya Ivanovich, and now by Abby Lott, Jenny Stevens, and Vas Mikopoulos. And it's one of the largest studies of inner city and civilian trauma in the country. And the numbers are astounding. And while we see this in Atlanta, we see similar numbers in Detroit and New York, parts of Milwaukee with Chris Larson's work, LA and others, in which we see rates of trauma on the order of 90, 95%. We see rates of people having seen somebody or know of somebody who's been murdered on the order of 40 to 50%. Approximately half interpersonal assaults, et cetera. So what we realize now is that our rates of PTSD in our at-risk communities is about 25%, is high or higher than in our military veterans. We really do have war zones. And that's very much contributing to our inner city and intergenerational cycles of risk, as well as the effects of structural racism on trauma and making it harder for people of under-resourced communities and of color often to succeed and break out of these communities. And so a great deal of work has been done over the last decade by my wonderful colleagues, Nagar Fani, Nate Harnett, and Sierra Carter and many others. This is just a few of them focusing on the role of structural inequities, both using the Grady Trauma Project and the ABCD consortia, looking at the effects of racial disparities, structural inequities, others on functioning PTSD rates and neuroimaging. So I'm not going to talk a lot about this today, but I wanted to make sure you're aware of the literature as we, as a field, try to bring together the work on understanding PTSD in traumatized communities, both from military, from war, but also from the effects of structural racism and cycles of violence. So now I'll start my talk with the background. An hour is a lot of time. Feels very luxurious. So how do I think about post-traumatic stress disorder in a big picture? So I've told you a little bit of background from some of the early, amygdala structure work, the community work, the treatment work. But let's back up until when we say PTSD, how do we think about this from a scientific perspective? So first of all, let's hold on to the general numbers of a trauma occurs and about 10% of people are at risk for PTSD, most recover. So that's the first thing. It's relatively rare, but not at all rare compared to like schizophrenia or other disorders. What are the different things that lead to developing PTSD versus not? So first is genetics. We know that about 30 to 40% of PTSD is genetically heritable, and that comes from both twin studies showing similar heritability in PTSD, depression, and addiction, and then now with very large-scale GWAS studies, genome-wide association studies that I'll show you in a minute. Why would a disorder of trauma, external environment, be genetically heritable? Well, it's not per se. The risk for how one responds or how one recovers from the trauma is what's heritable, and that's the same as we think about addiction. And then early environment. And then I'll talk a little bit about intergenerational environment as other potential predictors of risk. Importantly, early environment is childhood trauma. We know one of the biggest risk factors for all psychiatric disorders is childhood trauma, and certainly PTSD as well. And I'll present a little bit of work on some of the models that we can now develop about amygdala sensitization over the course of a lifetime due to early childhood trauma, and how that may lead to higher risk during later adult trauma exposure as well. This concept of early exposure and what the trauma is and how one consolidates the trauma is a very exciting one for me. First of all, we know that things like interpersonal trauma are much more higher risk factors for PTSD than non-interpersonal traumas. We think that is in part things like the break of trust in a relationship and things like that, and that some of our best protective factors against PTSD are social supports and how the interpersonal trauma disrupts that social resilience component as well. Also, the level of trauma. We think of the concept of trauma loading. Someone who's had 3 or 4 or 5 criterion A severe lifetime traumas is at much higher risk for PTSD than someone who's had a single one. And again, if one comes from a situation of historical trauma or ongoing violence, the rates of PTSD are higher. The idea of consolidation of fear. So new memories don't occur immediately, rather they are transiently activated, and this is whether it's an explicit declarative hippocampal memory or whether it's a threat amygdala Pavlovian memory. And what happens in the minutes to hours to days is different. You start with very rapid molecular changes, then nuclear transgenetic protein changes, new synapses, and eventually systems neuroscience changes with structural changes in the brain. And some of the work I'll show you related to sensory systems really makes very visually clear the sorts of structural changes that can happen in the brain with trauma sensitization. Then once one starts having trauma memories, that can push one down the path of increased generalization of fear versus discrimination of fear. Everything is safe, nothing, or no, there's some things, I mean, everything is dangerous versus there's some safety and some danger. Or sensitization, which we think is now really building on the concept of reconsolidation, where when a memory is reactivated, it's labile and can then be re-strengthened. And when I talk about some of our work with synaptic plasticity agents and D-cycloserine, one of the main questions, and this also I think relates to where we are now with psychedelics and ketamine, is if we can make the brain transiently more plastic, is that always good? And what we find in some of the work with memory reactivation is that it can both be resensitized or reconsolidated or be extinguished. We want to extinguish it. We want to train the brain to be newly safe, but there's a risk that we could strengthen the old trauma memory. So we'll talk about that as well. So in summary, some of the data I'll be showing, we'll start with genetics and early memory consolidation, how we understand early risk factors and how the early stages of trauma can lead to novel interventions. Then I'll talk a little bit about our work over the years beyond the amygdala, the sensory systems coming into the amygdala, concepts like the body keeps score, how trauma in the body, how trauma in the sensory systems may be increased sensitized and make this information coming into the amygdala more sensitive related to trauma cues. And then finally, how our understanding of extinction and then potentially cell type specific microcircuits in the amygdala may lead to novel ways of inhibiting fear. Okay, so again, we know from decades of work in the threat area, for example, more recently by Jim McGaw, but it goes all the way back to synapses that wire together, fire together, and early ideas of learning and memory, that memories are initially transiently encoded as short-term memories, then they are consolidated into long-term memories. They are then from that can be reactivated or reconsolidated into working memory. And then at that point, they can go down the path of repeat. And this is what we hope to happen in exposure therapy. You activate the working memory, you stay there. You're not getting the reinforcement that was expected. New learning occurs. And now safety memory is built upon this, et cetera. One of the places that we have great understanding at a molecular and cellular level, but haven't translated this into humans is consolidation. And the idea here is if we could identify ways of inhibiting the threat memory from being overly consolidated in the first place, could we have early treatments right after the trauma in the emergency department or on the battlefield to prevent PTSD from ever happening in the first place or ever developing? So in humans, we started this work because we really don't know much about the biomarkers of PTSD development. So parallel work has been going on for years now in terms of understanding memory consolidation in animals, particularly in mice and rats in amygdala function, as well as what's going on in humans. So I'm going to tell you a little bit about the work in humans. So the work that I've been involved in started with ourselves at Emory and Grady Trauma Project in collaboration with Charlie Nemiroff at Jackson Hospital in Miami. And this was a study where we identified people in emergency departments right after a trauma. It could have been a motor vehicle crash, a sexual assault, any other type of trauma you can imagine. And we were able to consent people while they were being medically cleared. And one of the cool things about this study is we were able to get IRB approval to add several experimental tubes to the stat pack that they use for cross and typing. So as soon as somebody got rolled in on the ambulance, we could get that early blood. And then we could do a whole host of questionnaires, physiological data, and then following them over time. The data from this led to a much larger study called the Aurora study that enrolled almost 5,000 people across initially about 20 emergency departments, led by Sam McLean at UNC and Kirsten Cohn and Ron Kessler and myself at Harvard. And the goal, and so I'm just going to show the very tip of this. Lots of papers have come out and we're still doing a lot of the analysis. I'm not going to show much about the digital phenotyping work, which is taking a long time to analyze, but it's very cool. So we had the Google Verily experimental watch on everybody right as they came in. We're able to have that for a year, in addition to a whole set of blood-based biomarkers, and then a subset of people got imaged at 2 weeks and 6 months. And so this is some of the early emergency department data. And first, what we would find is that people in 2 weeks after 2 to 3 weeks after the trauma would have a whole range of PTSD symptoms, some very low and some quite high. Most people would get better over time, but some stayed high and a few even got a little worse. So we saw some of the normal trajectories that one might see. And importantly, in terms of the circuits that we know are involved in PTSD, for example, amygdala hyperarousal to fearful faces with functional imaging, or anterior cingulate cortex, which often goes along with the amygdala in terms of, and the insula in terms of positive activation to threatening cues. Their activity at 2 weeks predicted 3 months and 12 months PTSD, suggesting this core circuit of overreactive threat was early on, even before we diagnose PTSD, one still sees a hyperactive threat circuit. In contrast, the hippocampus, along with the dorsolateral prefrontal cortex and the ventral prefrontal cortex, are generally serving as the brakes on the amygdala, if you will. And as we see hippocampal activation, that generally is associated with more extinction and less fear response. So we see an inverse relationship with hippocampal activation to cues. In this case, we're using behavioral inhibition tasks, essentially a go, no-go response. And when we do that, in the same 2-week period, high levels of hippocampal activation on the x-axis are associated with very low PTSD symptoms at 3 months and 6 months, and low levels of hippocampal activation when we should to a hippocampal probe are associated with high PTSD. So again, the inverse, the parts of the circuit that should be protecting the PTSD response are showing the same response a couple weeks in. In terms of developing new biomarkers, this is one of the ones we're most excited about because it's the most scalable and easy. So often while people were from a motor vehicle crash would be on a body board to clear their C-spine. They weren't doing anything. They were just waiting for the radiology. So if they consented, they'd allow us to talk to them. So they'd be sitting there and we have a portable galvanic skin response on their fingers and would do the interview. And if you simply ask open-endedly, what brought you into the emergency room? And you measure the galvanic skin response pre and post that question, it seems to be a robust predictor of who has PTSD 6 or 12 months later. So again, we're looking at the delta on galvanic skin response, which is essentially a readout of the sympathetic nervous system. Those who have high levels of activation when thinking about the trauma that just occurred are much more likely to have PTSD 6 months later. So as we talk about targetable subtyping, this could be an example of a scalable tool. Unfortunately, in the Aurora study, so this was from the original study, in the Aurora study, we did not see this level. It was slightly significant, but not robust. The main reason we think is because in the Aurora study, they actually didn't want to ask anything about trauma. So we can talk later about large-scale consortia and how you have to make negotiations and compromises. This is one we regret. And so we did the galvanic skin response, but it was during the demographic questionnaire, which isn't particularly emotionally exciting. And so we didn't see much signal. So yeah, as you would imagine, you have to activate the sympathetic nervous system to get the spread. So we're now working on further studies to see if this can be a useful biomarker when used the right way. Okay, so that's some of the early data on consolidation. We've got data on galvanic skin response, wearable devices showing lower heart rate variability with PTSD, a number of other imaging studies showing activation. And I'll come back in a little bit to show some of the data on the visual system sensitization associated with later PTSD. But I want to show a little bit on the updates from the genetics. So in terms of the broad scale risk. So one of the risk factors for PTSD is what are the differential biological mediators and markers of consolidation. The others, of course, is the genetics that you bring to it. And so excited to announce coming out, I think tomorrow in Nature Genetics is the largest genomic architecture of PTSD from the Psychiatric Genomic Consortium PTSD working group. So this is led by Caroline Nievergelt, Kirsten Conan, Murray Steen, and myself. And this combines about 50 different consortia across the world, including now the Millions Veteran Program, the UK Biobank, and a lot of the small studies. So Kirsten, Caroline and I are the co-leaders because our cohorts in about 2005 started the consortia. So the Grady Trauma Project, and what's nice about the PTSD consortia, it's also one of the most diverse in terms of ancestry of all the PGC, both in terms of our early work in Grady, some of the later work in Detroit and other diverse cohorts, as well as a lot of work in Africa from Kirsten. And we now have over 1 million people in this and about 200,000 cases. And for a number of years, schizophrenia was showing Manhattan plots like this, and we were all very jealous. So we're now to the point in PTSD and depression that we really have large scale GWASs of common variants. So is everybody familiar with how to read a GWAS, or should I do a one sentence? Okay. So what we're measuring here is essentially every dot is a single nucleotide polymorphism. That's an AZ, G, or T. And the ones that are chosen, while there's millions of these in the genome, we're probably hundreds of millions. The arrays for these are basically finding the ones that are common. And so that have variation, usually around 3 to 5% or more. And each dot is essentially a t-test for the A version or the C version, for example, about does those two different versions predict the outcome of interest. In this case, it would be PTSD diagnosis, which when you're looking at 1 million people is defined in a very heterogeneous way. So we can talk all about the caveats of this. And then because you're doing essentially 1 million analyses, you have to have a multiple test correction for 1 million. So 0.05 divided by 1 million is about 10 to the -8, and that's where the genome-wide significant line is. So we now have about 120 genes that make up about 80 to 90 loci, and there's a number of really exciting ones, including the beta-catenin system, FOXP2, which is associated with language, but also is related to amygdala function and the intercalated cells that surround and inhibit the amygdala. FOXP2 is the main marker of those cells, and you actually see changes in FOXP2 expression with threat. The GABA-R1 receptor, so one of the main GABA receptors, a number of NMDA receptors, CAM kinase, a main marker of plasticity and pyramidal cells, the estrogen receptor, and a number of other really cool things. So we're excited about this. There's a high overlap with depression, but there's also quite a few that are non-overlapping with depression, and diving into both what's shared versus what's distinct will be really one of the fun things for next steps. But one of the genes that came up in the first freeze that continued to be strong in the last freeze is the one I'll focus on. And that's Ned and Maya's favorite gene, the corticotropin releasing hormone receptor. So again, remember that the canonical stress axis is the hypothalamic pituitary adrenal HPA axis. At the top of that is CRH, which is released from the amygdala and the BNST, and both activate the hypothalamus. The PVN releases CRH onto the pituitary, causing the adrenocorticotropin releasing hormone, ACTH, to be released, leading the adrenal gland to release cortisol and adrenaline, epinephrine. And that's the classic loop. I'll talk some about how cortisol feeds back on the system. Across studies for decades, starting with a lot of Rachel Yehuda's work and others, suggested hyperactivity of the cortisol system, and then also a hypersensitivity of GR feedback, so that in classic PTSD, without dissociation, without depression, you tend to see rapid responses of HPA axis and very rapid shutdown of the HPA axis after dexamethasone. Whereas in depression, you might see sort of a blunting or a GR non-responsive response to stress. So across these multiple genes, there are several that are related to HPA and stress regulation, and quite a few related to synaptic plasticity. So those are two of the themes I'll talk more about. In terms of CRH and the role of CRH in the basic circuits of threat and fear, and how might genes in the CRH receptor and CRH axis be related to threat and fear, there's decades of studies, because again, this is the area we understand. I'm getting called by spam. So I'll just show a few of them to support the general concept that genes related to PTSD in humans can also be related to threat markers in rodents in a translational way. So this is work from our group collaborating with Charlie Nemiroff and others at Emory about 15 years ago, showing that if we overexpress CRF in the mouse amygdala, we could get increased HPA axis, increased cortisol, but importantly, increased baseline and threat-related startle responses. And that's one of the core things we see in PTSD, this classic startle response. If we manipulate it in other ways, in this case, we're overactivating the CRF circuits by knocking out the GABA A receptor. So if we knock out receptors related to GABA, and remember one of the GABA B receptors is also involved in the survived genetics in the PTSD cohort. But when we knock out GABA in CRF neurons, we make those cells hyperactive at a physiological level. And so when these cells are now physiologically hyperactive, we're unable to be suppressed by GABA, you see increased CRH expression, increased cortisol release, but importantly, increased consolidation of fear and deficits in extinction fear. So after Pavlovian tone shock pairing, so this is 5 tone shock pairs during acquisition of fear. The two groups, the GABA knockout and the control, look the same. But during expression, those in the first trials, those in which we've knocked out GABA, the inhibition on CRF cells, show more fear. They've over-consolidated the initial fear. And importantly, they stay afraid. They don't extinguish. And again, this is what we see in patients with PTSD. They have this fear that is deficit to extinction. Now, this by far is not the only story, but it's an intriguing one to combine as part of the pathways, at least in which some of the common variants can support some of the more mechanistic work at the brain level. I think Elizabeth was here last year or 2 years ago. So Elizabeth Fender, who is now the director of the Max Planck in Munich, and has been a colleague for about 20 years, and is just a wonderful person, though she was snowed in during my ACMP special talk that she was supposed to give in Munich. So she and I showed first that FKBP5 was related to childhood trauma. So let me walk you through this. So I've talked about CRH. And when cortisol feeds back on this system, multiple levels at the hippocampus, the amygdala, the HPA axis, and the hypothalamus, cortisol binding to glucocorticoid receptors both activate stress-related genes, but also shuts back down this system. What we see with PTSD is a hyperactivation of the system and a hypersensitivity, so that when that cortisol more rapidly shuts down gene expression at these levels. So what could cause glucocorticoid receptor hyperresponsiveness or hypersensitivity related to PTSD? And for decades, the FKBP5 gene has been a very interesting candidate. So FKBP5, I'm not talking a lot about it, so I don't have the background slides, but it serves as a protein that shuts down GR responsiveness. It's involved in allowing the cortisol to bind with the glucocorticoid receptors, translocate into the nucleus, and activate gene expression. More FKBP5 prevents GR translocation. And what we showed was that two different polymorphisms of the FKBP5 gene were differentially related to PTSD function. So that there seemed to be one in which, on average, if you look at childhood trauma, everybody with more childhood trauma has more PTSD. But if you break this down by genetic FKBP5 allele, there seems to be a more risk allele and a resilience allele, so that a subset don't seem to be sensitive to increased levels of childhood trauma. The risk allele, on the other hand, is associated with more PTSD, more amygdala activation, decreased hippocampal functioning, and differential methylation. So there's a whole story that I decided not to talk about because I want to talk about other stuff, but it's another area in which we're seeing HPA axis support at a genetic level and at an amygdala level. I'm going to summarize all the HPA stuff with this model. That the amygdala is constantly taking in information from the world around us. Is it a stick or is it a snake? My wife's amygdala thinks it's a snake. I think it's a stick. I go down the resilience tolerance. I don't care about this path. She goes down, I'm afraid of this path. But relatively quickly, her hippocampal and prefrontal kick in and sort of shut it down. Say, okay, it's just a stick. It'll be all right. Childhood maltreatment, poverty, violence, exposure, structural racism, and others, we think lead to an amygdala-dependent emotional circuit that's always primed for stress responsiveness, a hyperactive amygdala system. How might that occur? Well, people like Regina Sullivan have shown that developmental cortisol leads to hyperactive amygdala and more rapid maturation of the amygdala in rodents. I think our work shows that FKBP5 is one of the mediators of cortisol sensitivity, leads to differential risks for that developmental cortisol, depending on how sensitive the GR system is to that developmental cortisol. So that in the wrong combination, I guess, of genetics, with one example is FKBP5, but of course, there's hundreds of these genes that are acting in different ways on this, combined with early life stress, lead to an amygdala system that is always primed for stress, fear, and I'm going to posit next in potentially the fight part of fight or flight response that we don't generally talk as much about. So most of our work over the years has been focused on threat responsiveness, and I wanted to shift gears a little bit and talk about the cycles of violence and threat responsiveness. And from some of the early work at Grady, in this study, I think there were about 4,000 participants. Even when we controlled for sex, age, race, education, employment, income, substance abuse, we still saw a very strong association of PTSD with having been arrested for a violent offense or behavioral relationships to violence. And it suggests that untreated PTSD is part of the cause of this. And how might that occur? This is a follow-up study in which self-report of violence-related behaviors was highly associated with PTSD, untreated PTSD. Emily Newman is a senior postdoc in our group who's been looking at the role of CRH in stress response, and it's particularly in threat-related, stress-related aggression. And so in the central nucleus of the amygdala, so this is that same region that we've been talking about, leading to multiple brainstem projections related to the threat response. It also projects to the PAG and other areas that have been known to relate to aggression. And so Emily's been using calcium imaging specifically in the CR8 cells in a intersectional virus approach, in which we specifically can look at calcium imaging with in the central amygdala CRF cells. And she looks at aggression in two different ways. One in which the animal is being attacked and just basically has a self-defensive fight, and this is essentially a control situation because the animal's not initiating aggression. It's just in a self-defensive behavior response. The other is a group of animals that have been socially isolated and other stressors. And when they have had these prior stressors, they are now more likely to initiate an aggressive attack. And what's interesting is when we're looking at calcium imaging of the central amygdala CRH cells, the defenses, so essentially just the automated self-defensive response, don't activate these cells at all. But whether if we're looking at an offensive attack to an aggressive opponent, so again, it's a previous stress-related aggression to an animal they're afraid of, that gives you the largest signal of CRF cells. And that CRF signal can predict 5 to 15 seconds ahead of the aggression. So they'll be on this part of the cage and go over there and it gets activated. And we see a nice correlation between the peak size and the prediction of the aggressive response. So that's what the chemogenetic correlational data are suggesting, that these CRH cells, in addition to lots of other things we know about these CRH cells, are predicting the aggressive response. So then the question was, can we block this causally by using chemogenetics to give an inhibitory dread that would inhibit CRH activation, and would that block aggression? And hopefully this will work. So on the left, you're seeing this is the normal aggressive response of a prior stressed animal. Could you play that one more time? On the left side, and then we'll play the right side. Okay, so it smells it goes up, and pretty much up in here, it would start to know. And this is all combined with Sam Golden's deep lab cut and Simba analysis. And then on the right, these same animals have been given DCZ prior to having an intruder. And you can see it has what appears to be all the same exploratory behavior, but that exploratory behavior never shifts to aggression. And she's repeated this quite a few times now. So can you go on to I guess I can go to the next one. So the take-home message here is, if we look at CRH inhibition, it doesn't alter the approaches. It doesn't alter the contacts, but it essentially completely eliminates the attacks right before. So she's also showing now that if they overactivate it with a GS, they get unexpected aggression, much increased aggression, and they'll even attack females. So it's an interesting connection of this classic CRH system that we've all studied across species as maybe also being rolled in the fight component. So the summary here is a subset of CRH cells in the central amygdala are active during social investigation of an intruder. That leads to cell activity predicting transition to attack, and that CRH activity is necessary for the transition from the social context to the attack, and supporting the hypothesis that these cells are important for the fight component of the fight, flight, or freeze. Interestingly, there's been a lot of CRH data related in postmortem studies, as well as spinal fluid related to aggression, to suicidality risk. And I'm not going to talk about it today, but it might be fun in the discussion. One of the components of one of the debates in suicidality research is the difference between suicidal ideation, which is quite common, and actual suicide attempts. And suicide attempts seem to have a component of negative emotion that's related to ideation, but then a component of impulsiveness and aggression to it. And so one wonders, and we can also talk in the discussion about some of the failure, if you will, of the CRH antagonists in clinical studies. And we all believe this because we haven't known who to treat with the antagonists. And is it something about this sort of aggressive, impulsive component that could be critical? Okay. So I'm going to move from the amygdala and the classic systems to some teaser data related to other sensory systems. So PTSD, long before we thought about it as we do now, and again, PTSD has been talked about in the literature really since the ancient Greeks and Romans. And the idea there was that something would happen to someone, whether it be they'd be in a fire, they'd be in an attack, they'd see someone killed, and they had numbness, or it was almost like the Freudian hysteria, or they were changed forever, but there was no effect of physically on their body. Some of the terms were shell shock, soldier's heart, neurocirculatory asthenia, battle fatigue, et cetera. So for a long time, we've known about these components. And in the literature, Bessel van der Kolk's book, The Body Keeps Score, is a relatively famous book about the fact that the field hasn't really dealt with the problem that patients with PTSD often feel the trauma in their body. What does that mean to feel it in your body? Well, it's probably not in our body, but it's probably in the brain representations of the body, right? So I'm going to show brief data on structural changes in sensory systems following fear and threat across the auditory circuit, the olfactory circuit, and the visual circuit. And the hypothesis, and then this builds upon a long history of sensory system changes that we've known can happen certainly developmentally. And for a long time, it was thought that sensory systems have early critical periods, and when those critical periods close, there's no more plasticity. But increasingly, we realize that there's transient openings of plasticity all the time. And people like Mike Merznick and John Cass, Michael Streicher, Weinberger, and others have shown that with that these periods of plasticity can be opened in sensory systems. And so our question is, can these sensory plasticities be opened with trauma and relatively low levels of trauma, as I'm going to show you with Pavlovian conditioning? So our first experiment, this is from Orion Kiefer at Emory in about a decade ago now. He was particularly interested in we had a great 9.4 Tesla magnet at Emory that we could scan up to 16 mouse brains in vitro, ex vivo, at the same time. And the question was, we see voxel-based morphometry in structural studies all the time, but there's very little studies that really get at the question of what's really going on with the increased gray matter density in voxel-based morphometry. So Orion did a series of auditory fear conditioning, where they got 10 tone shock pairs over multiple days. So it was much more fear conditioning than normal. We had no idea what amount we needed. But when he did that, not only do we see very robust fear in these animals, with voxel-based morphometry, he saw increased gray matter density in the threat circuit. Insula, basolateral amygdala, central amygdala, medial amygdala, but also primary auditory cortex. And what was special about this is the same animals, brains that were going through the ex vivo study also had the Thy1 gene. And Thy1 is special for a lot of reasons I'll talk about later in terms of its amygdala expression, but it also expresses in a subset of sparse pyramidal neurons. And so he saw increased VBM across these brain regions. Then when he did spine analysis based on the Thy1 expression, he saw very robust increased spine density in auditory cortex. In these other regions too, that was the best place to focus for the spine analysis. And with Scholl analysis, he saw that increased spine density several weeks after auditory threat conditioning was highly correlated with the increased voxel-based morphometry. And even when we saw no changes in cell nuclear density or in glial density, and that the increase in spines accounted for probably about 50 to 60% of the increased voxel-based morphometry effect. Okay, so that's one example of auditory cortex. We've shown other studies that if you block auditory plasticity, you can block that conditioning. I've been talking way too much, and now I'm going to have to get through a lot of slides quickly, so forgive me. The olfactory system is exciting because we know specifically the neurons that each neuron that expresses a single olfactory receptor type is unique to that receptor, and all of the neurons in the nose that express a specific receptor converge to a specific glomerulus in the olfactory bulb. Olfaction is one of the most emotionally sensitive components of sensory systems, and people who have an olfactory component to their trauma, that's often some of the most refractory and also strongest for triggers. So Seth Jones in our group had shown in 2008 that if you do olfactory fear conditioning, so olfactory foot shock training, and wait a couple of weeks, you get robust increases in number of neurons in the nose and the size of the glomerulus. So in an adult animal, the olfactory system is plastic. Philomene Morrison went on to show that you could replicate this with odor shock pairings and get increased number of neurons in the nose, increased glomerular size. But interestingly, if you then extinguish the animals so that they're no longer afraid of that odor and wait longer, that reverses the number of neurons in the nose and reverses the size of the glomerulus. So at the base in adults, the olfactory system is plastic and can be increased sensitive based on, we also showed if you did cocaine place pairing with odors, you get the same thing. So it seems to be valence independent. So one of the questions we all have is, what is this concept of intergenerational transmission? We know that if your parents have PTSD, you're at a higher risk for PTSD. We don't know if that's because you observe your anxious, your parents who are fearful. We don't know if it's because you grow up in a similarly risky environment, if you have shared genetics with them, or if there's something else, possibly epigenetics. This is one example of physiological intergenerational inheritance at Grady. If we measure children's startle and ask about their mother's history of abuse, mom's history of abuse is associated with much more startle than moms with low abuse. So Brian Diaz asked, could we use the olfactory system as a model for intergenerational transmission? And basically it worked remarkably well. So we trained the fathers to be afraid, and then we waited 2 weeks, and then we mated the fathers. And this is the offspring, the F1 offspring. So the F1 offspring of home cage controls had normal levels of axons for this receptor and normal glomeruli. If they had a different odor, propranolol, that doesn't activate this receptor, but they're equally afraid of propranolol, you don't see any change in this system. But if they've been trained to be afraid of the odor that activates these receptors, their offspring, despite never having exposure to this, show the structural changes of increased sensitivity to that odor. So this just quantifies it lots of different ways. It holds across grandparents, so it goes at least one generation down, even though the intermediate generation never had exposure to the odor. And we can see it go in both directions. So in addition to the increased glomerular size, there's increased startle to the trained odor in the grandchildren, whether they were trained to acetophenone that activates this, or if the grandfathers were trained to propranolol, they show no more behavior to propranolol. And importantly, with this, we were able to do in vitro fertilization, which is why we did it in the males. So if we take the sperm and have them create new offspring via in vitro fertilization, where the animals never have exposure to odor, we still see the increased glomerular size, suggesting that there's something in the broad term of epigenetics that may be mediated because there's no ability for the animals to have any behavior or interaction components with this. And we were able to see some methylation changes in this odorant receptor in the sperm, though the mechanisms and everything else of that still need to be worked out. Okay. So finally, the third example, so olfactory system and auditory systems. What about visual system? And we have done less of this in the rodents, but in the humans, the data have been really robust. So in our so I started with just showing auditory visual cortex. Of course, a huge amounts known about the visual system and how critical this is across all species, but particularly in humans. Nate Harnett's work initially with the secondary analysis of the Grady Trauma Project study, and then I'll show you in a minute the Aurora study, showed something that many of us often see in the threat field is we see a lot of visual cortex activation and we usually ignore it. But when he dived into it and he did multimodal analyses combining structural analysis, DTI, VBM, as well as functional analysis, over and over, so he sees increased visual cortical structure and functional activation related to PTSD development over time. And then was able to replicate this in the Aurora study, that structural covariance networks associated with increased visual cortex representation were associated with increased PTSD symptoms at 2 weeks and later. And finally, these same covariance networks were associated with increased intensity of nightmares and severity of nightmares, raising the question of, is what we're seeing in primarily motor vehicle accident traumas, which there's a huge visual component, either a change in visual sensitization after the trauma, or does this somehow represent people with prior traumas or increased visual sensitization that made them more sensitive to the visual component of the trauma at the time? And we don't know that yet. But together, this work starts to suggest that there are sensory structural changes in sensory systems upstream of the amygdala, and that neural processing in sensory systems likely represents a critical component of sensory-related threat encoding. And this may underlie pervasive components of sensory intrusive memories, sensory triggers, and sensory-related nightmares. All right, and my last part, I may go a couple minutes into my discussion, but not much. Is what can we understand about inhibiting fear and how will that take us to new directions? So I started with this simplistic model of exposure therapy, right? And this maps onto our model of Pavlovian extinction. Repeated exposure to a conditioned stimulus without reinforcement from the unconditioned stimulus leads to the brain relearning that that conditioned stimulus no longer is predictive. And what happens in threat is that essentially you're learning a new safety memory, and we can talk about how we may know that from a behavioral perspective. But we also know it's extremely plasticity dependent, and one of the most well understood genes related to plasticity is the NMDA receptor, and the amygdala and the hippocampus are two of the brain regions that are most chock full of NMDA receptors. Mike Davis's group, who's really one of the titans and grandfathers of the amygdala field, showed us that learning to extinguish fear is an active NMDA-dependent process. So what do I mean by that? These are rats who were trained to be afraid of lights. They were every time they were put in a dark box and gave a startle cue. And every time the light came on, they would get a shock. So that now the animals are afraid of lights. And now when we turn the light on, they startle more. If you now give them the lights in the absence of any shock, so 60 lights, that's essentially an extinction curve. And this is what happens. They're no longer afraid. That's exposure therapy for the rat. If they give them the same 60 lights in the presence of a drug that blocks NMDA, AP5, they're just as afraid as they ever were. So learning to inhibit fear is an active NMDA-dependent process. In Dave Walker and Kwok-Sung Lu and myself and Dave's and Mike's lab a decade later showed that if you gave a drug that enhances the NMDA function, D-cycloserine, that activates the serine glycine moiety to make calcium flow through more likely, you can give a partial extinction and you get as much extinction as you do if you gave twice as much exposure. So by enhancing plasticity, you can make extinction work better. This was exciting because D-cycloserine was already shown to be safe in humans. So we were able to rapidly translate this. And this is in 2004 with Barbara Rothbaum, who is one of the leaders of exposure therapy. And this is a virtual reality exposure therapy study where people went up virtual heights and got exposure to the virtual heights, either with placebo or D-cycloserine. And normally this would take 6 or 8 sessions to reduce your levels of fear by 50%. And in this case, only 2 pills in a placebo-controlled fashion reduced it as much as normally 6 or 8 sessions. So we showed that we could enhance extinction in humans just like we could enhance extinction in rats. And that was really exciting. And the psychology field got very excited because this is the way, really one of the first ways of specifically using a cognitive enhancer to enhance psychotherapy. And so in social anxiety, Adam Guastella in Australia, Stephen Hoffman in BU, in OCD, Sabine Wilhelm, Matt Kushner, and in PTSD and panic disorder, people were seeing nice, rapid effects of increasing exposure therapy. As these studies started growing, this failed to replicate, as with many things that are exciting when they start. And we were not sure as a field, was this because people were sourcing the D-cycloserine from different places? Is it because we're using different doses? Is it because we weren't giving it at the right time? Or we weren't knowing how to use the therapy? Barry Everett, one of the leaders in learning and memory about that time, had a very interesting paper. If you remember, I talked about sensitization as reconsolidation versus extinction. Turns out if you reactivate a memory, and you can do this in a rat as well as you can in human, but instead of giving, say, 100 re-exposures, you only give 1 or 2 re-exposures. That activates the reconsolidation. You reactivate that engram. They showed that D-cycloserine enhanced both extinction and reconsolidation depending on the length of the extinction reactivation session. So if you give a short exposure, you can enhance reconsolidation. If you give it long, you enhance extinction. In Adam Guastella's data, they post hoc analyzed it, and patients who received D-cycloserine, if they had a good within-session extinction, they got much better faster. If they had no within-session extinction or even got a little, or even showed a little, increased anxiety, they actually got worse over time. So that was at least consistent with that. So it suggests that until we get better control over what for any given individual is driving extinction versus reconsolidation, we're at risk with synaptic plasticity. And as more recent experiments have either combined D-cycloserine with other drugs to sort of push the valence or give it different ways, it's looking more positive, but it also makes us worry, or at least be aware of, with things like ketamine-assisted exposure therapy or psychedelic-assisted exposure therapy. We're enhancing plasticity, but we're not necessarily driving the valence or driving extinction. And so we have to be careful there. And so in the future, where we would like to go is ask the question of, could we enhance plasticity in a valence-specific way? Could we use science to drive plasticity of extinction and not be at risk for driving plasticity of sensitization or reconsolidation? And a number of people, including Andreas Luthi in Basel, showed that there are different pathways for both fear on and fear off within the amygdala. In this case, they recorded from amygdala basolateral neurons, and they found about 30% of them responded to a tone during tone shock conditioning or and after tone shock conditioning. They called these the fear on cells. About another 30 or 40% of them they called extinction or fear off cells. They didn't care about the tone before training. They didn't care about the tone at fear conditioning. Once the tone was extinguished and no longer fearful, they then responded to the tone as if these cells came on to inhibit that threat response. If we knew what these cells were, could we target those in a genetic or pharmacological way to more specifically enhance extinction over reconsolidation or sensitization? I've talked about the amygdala, and now we know there's about 15 subnuclei, and now we know there's hundreds of subtypes. But now with single cell sequencing and other tools, we can really start to dissect these. And I'm going to end with just talking about one set of these cells, which is Thy1. And so Thy1 is a developmental marker. It's been used a lot, as I showed you earlier, in some of the sparse pyramidal neurons. But interestingly, it's expressed in the most dense in the brain in the amygdala. And it's expressed in about 30% of the CAM kinase positive basolateral amygdala neurons. It's thought that the amygdala development comes from both a subcortical and a cortical developmental pathways. And it's thought that the Thy1 may represent the cortical. And interestingly, as opposed to the canonical story where BLA cells project to the central amygdala, none of the Thy1 pyramidal neurons project to the central amygdala. They instead project primarily to the accumbens, and they project primarily to the infralimbic over the prelimbic. So it seems that it's marking a subset of projection neurons. When you activate these, it inhibits the physiological flow through from the lateral to the central. And furthermore, when you inactivate these with optogenetics, you get a blockade of fear consolidation and an enhancement of extinction. And you can do that with optogenetics or chemogenetics. So it looks like we're to the point that we can start to target specific subtypes or microcircuits within the amygdala that are more fear off compared to fear on. If we take that to the next step and say, if we identify cells, in this case, Ken McCullough did gene expression analysis specifically of these Thy1 basolateral neurons and found a number of really interesting targets, particularly neurotensin 2 receptor. And neurotensin's been implicated in the psychiatry field for a long time. But if the neurotensin 2 receptor in the amygdala is specifically in the Thy1 neurons and not the other neurons, so potentially in the fear-off neurons. Neurotensin 2 is a GS stimulatory G protein. There's never been a study looking at neurotensin 2 activation, but the argument was if you use an agonist and you activate the fear-off neurons, you would pharmacologically turn off fear. And that's exactly what we saw, a robust inhibition of fear and enhancement of extinction of fear based on our understanding of the microcircuits and the neuropharmacology of that microcircuit. And then this was later expanded by work from Kate Ty's lab that we collaborated on, showing that the neurotensin can push and pull positive and negative valence within the amygdala. So our goal then is we're starting to reach the pivot point where a whole host of new targets can be identified based on our understanding of the neuropharmacology and the microcircuits and specific cell biology in the amygdala, that we might be able to have new treatments of chronic PTSD, but also ways to intervene and prevent short-term consolidation. I talked briefly about genetics and consolidation, what's upstream of the amygdala in terms of sensory sensitization, and how understanding extinction could lead to novel interventions and treatments. The simplest take-home message is that the amygdala is always being modulated by these other areas. The brakes have essentially fallen off in PTSD. Our trauma-informed treatments currently enhance top-down regulation of amygdala. Our current drugs essentially just decrease overall symptoms, but don't target the actual memories. And we hope that our future neurobiological-driven approaches can really target in valence-specific ways memory enhancement. And I will stop there with a lot of other text you can look at while we talk, and making the comment that a precision medicine approach to trauma and psychiatric disorders is feasible based on advanced understanding of neurogenetics and circuit dysfunction. Thanks so much for your attention.
Thank you so much, Kerry. Great way to begin. And I'd like to now invite Estelle Higgins and Hadley Rarig up to begin the questions. Estelle is a graduate student in psychology and Hadley is a postdoc.
Oh, I thought they were skipping out on this. So do we all sit here? Okay. I hope that made sense. I chronically try to jam too much in a talk, and I'm sorry. Okay, I'm just going to keep using my lavalier. Okay.
Very Thank you so much. These are some fascinating ideas, and especially your work with transgenerational transmission of fear and in the sensory systems. So I'm interested in sort of this idea of transgenerational fear transmission, but could we also envision a similar process of transmitting resilience transgenerationally, and how might these mechanisms be different or similar, and would you anticipate that upstream systems would also be involved in that kind of transmission?
Yeah, great question. So one of my favorite studies I didn't put in, but after the work that I showed with Brian, in his own lab, they showed that if you fear condition the mice, the fathers, and then extinguish them before you mate them, the offspring of the extinguished fathers, their level of olfactory sensitivity and olfactory sensory changes looked like untrained animals. So that at least at that level, in the same way that I showed in the PNAS paper, that in an adult, if you fear conditioning and then extinguish, the olfactory system goes up and down. So whatever is happening to transmit this across generations seems to be at some level in real time sensitive to the animal's state. So if we can extinguish it, that would suggest, to your point, that there might be ways of training resilience as well if we had a better handle on what it is that we're training. I think the other caveat of that is, I don't think this is going to hold up across everything. There's, if biologically this is real, there's probably aspects of biology that can be transmitted and probably aspects that can't. A lot of work by Tracy Bale and sorry, my name recall today is slow, so I'll be a little, but for folks in Europe have shown that general stress sensitization changes next generation phenotypes, but it's been really hard to nail down what that is. But you could imagine that there might, at the genetic level, there might be HPA axis signals that could be transgenerationally transmitted. The olfactory system, because the genes are actually encoding the receptors, it's easier to understand that. It's harder in other sensory systems that are really not encoded at a genetic level to know how that would be transmitted or at more complex brain function. So my guess is there are going to be certain areas maybe that we call instinct that are able to be encoded in a relatively simplistic way around sensitization or something like that. And then there are probably going to be other things that can't. But that's a guess more than data-driven.
Thank you, Sam.
Hello, thank you for the great talk. I was hoping you could speak towards some of your research showing that women are predisposed to greater PTSD risk. So specifically, how do you think sexual dimorphic mechanisms underlying stress-related physiology may operate on a developmental timeline? So, through early childhood development, adolescence, or postmenopausal adulthood. And how might sort of these developmental interactions affect the presentation of PTSD symptoms, do you think?
Great question. Probably hours of potential responses there. I'll give one specific example that I didn't show today. So one of the areas that we've done a lot of work on is called PACAP, or pituitary adenoid cyclase activating peptide. And PACAP is both upstream and downstream of CRF. It's another stress peptide that in both the amygdala and the hypothalamus is very critical. And we showed that PACAP polymorphisms were associated with PTSD, but particularly more so in females than males, and that PACAP levels in the blood and PACAP genetics were associated more with female than male. And specifically, we were able to then show that in the PACAP polymorphisms in the receptor that looked like sexually dimorphic, if you then do cell culture models, that there's actually estrogen response elements upstream of the PACAP receptor, and that polymorphism moderated estrogen receptor binding. And so that one mechanistic way that we were seeing it was actually estrogen receptor differential sensitization of the stress pathway. So that's one mechanistic story. The fact that the estrogen receptor is showing up on the GWAS, we don't know what that means yet. But it's interesting. There seems to be differentials. We know even when you control for sexual assault, which is the most obvious sort of first sexual difference that we see, that it appears even with things like motor vehicle accident that you see increased risk of PTSD in women compared to men. In the general psychiatric sexual dimorphisms, we see about a 2-to-1 or 50% increase in schizophrenia and autism spectrum disorders in males, and about a 50% increase in affective disorders and anxiety disorders in females. And there definitely appears to be, I think the field hasn't fully separated out the developmental trauma component from that, as well as the developmental trajectories of sex hormones during puberty. But it's thought that all of that's really critical.
Thank you.
Yeah, thanks.
Should I go again? Okay. So another thing that I'm interested in is sort of behavioral interventions. And how can we take what we are learning about these molecular and physiological changes to create or augment behavioral interventions, especially when you're thinking about sort of the level of specificity when it comes to plasticity in terms of extinction versus potentially enhancing fear memories.
Again, how do we do it? So, I don't think we know the answer yet, but what I do hope, and I think where a lot of excitement's going, is how can we get more quantitative and predictable tools, right? The field of cognitive behavior therapy is long and vast and has been quite successful. And yet, for the most part, you're still using subjective units of discomfort. How anxious am I on a 0 to 100 scale to kind of drive everything? We should be able to do better now, right? And so I think grants that are looking at, really measuring physiological digital phenotyping. I'm sure some of the other speakers are going to talk about digital phenotyping, including Dr. Ressler. But you could imagine that if we were doing the part of the problem, and Nafoa's work had shown that within session extinction does not predict between session extinction recovery, but there's a lot of nuance to that. And then separately, our decycloserine, the Gorcella study suggested that within session extinction while on decycloserine was big difference. But again, that within session extinction is only measured by self-report. As we'll hear throughout the day, self-report is very problematic. Since I may not have another chance to say it, I'll say my favorite metaphor. If you went to the doctor 100 years ago, they could use a stethoscope and tell you where your cough was, maybe which lobe it was, and whether it was wet or dry, and probably a few other terms that don't really mean anything. And then they could give you codeine and you were cured, for a little while. You could argue that the monoamines, that most of our current psychiatric drugs are kind of the codeine of the brain. They're sort of in a non-specific way decreasing some of the responsivity. But until we really understand the biological differences between different subtypes of lung cancer, heart failure, autoimmune pneumocystis, et cetera, you can't target the underlying biology. But it all feels like a cough. What if our experience of anxiety or some of these negative emotions is not any more biologically based than our experience of a cough, right? We have thousands of ways the brain can break, if you will, to give this experience. But if we're saying on a 0 to 100 scale, I feel anxious. So we've got to have the better biomarkers to give us much more quantifiable and reliable markers to then iterate and then to really subdivide better what we're targeting with our behaviors. Yeah.
I have another question. I'm interested in some of your research that's demonstrated that PTSD impacts downstream systemic inflammation. Does systemic inflammation likewise exacerbate symptoms of PTSD, possibly through increased neuroinflammation? Is there evidence to suggest that there's maybe a bidirectional relationship between trauma expression and inflammation?
Yeah, great. I'm very impressed. You went none of the things I actually talked today, but thanks for asking about. So it's great. Yeah, so we've got data in the emergency department, for example, showing that there's increased CRP levels associated with later PTSD, and a whole host of data in the PTSD literature from us and many others that look very parallel to what we see in the depression literature, that you see increased inflammatory cytokines, decreased suppressive, immune suppressive cytokines. What does all that mean? We don't know. It's the simple answer. the hypotheses are that it goes both directions, right? That dysregulation of the HP axis. So one simple hypothesis is that if you have super sensitivity of cortisol and you actually have less cortisol, you have less endogenous suppression of the HP axis. And if you have hyperactivation of the adrenaline system, you have hyperactivation of the HP axis. That's one of many examples. But almost certainly the glial and the microglial story is really critical. We have a recent paper looking at the intersection of stress with microglial activation and what's called secretory autophagy. So when neurons release exosomes that have a whole set of cytokines such as TNF, IL-6, IL-2, and others, that activates microglia locally, and that genes involved in that are also involved in cellular breakdown and dementia. And so I think the field's starting to get at the local microcircuit level more indication of where cellular stress response is intersecting with microglial stress response and how that may also lead to stress integration with things like neurodegeneration. But where the brain body part, I think there's really interesting work in the field about bodily signatures of both inflammation as well as metabolic cues impacting the brain as well. But I think it's only really the tip of the iceberg.
Thank you. That's super interesting.
You can probably yell. Oh, I guess the Zoom people can't hear you if you yell. Okay, great. I can repeat your question if you want to start now.
Thanks, Amber. First of all, thank you. It was just fantastic. The data you presented on sensory systems, when you introduced it, you linked it to Bessel's work and the phenomenology of PTSD in experiencing bodily symptoms. That's where often these symptoms are phenomenologically experienced. And of course, you made the point that they may not be experienced literally in the body, but in representations of the body in the brain. Of course, one major area where that kind of processing occurs is in what we think of as interoception. Yeah. So I wondered whether you have sort of focused your analytic lens at all on interoceptive processes as another potentially major domain that might be implicated in some of the specific symptomatology of PTSD.
Yeah. I don't know if you guys have had Saib Khalsa give a talk here yet, but Saib's one of my favorite people, and he's becoming one of the leaders in interoception. I'm a huge fan of it. We've not targeted it directly. Certainly, we see insula in a lot, and I think trying to figure out when anterior versus posterior insula, what is it doing? It's kind of like the anterior cingulate. It's kind of involved in everything. So trying to parse that out is hard. In that sort of first voxel-based morphometry paper, we saw insula just like we did with auditory cortex. And we do have some insula studies in mice that we're getting off the ground where we both certainly see it with threat, but we see it a lot with threat generalization as well. But I think the question of how, I'm just trying to think of how either at the human level in correlation with seeing insulin in a lot of things, or more at the animal level in terms of trying to get at causation and mechanism, even what the experiments would be, is the model that the insula is maybe in part mediating some of the primary and higher order sensory interactions. You could imagine, say, if you were to do functional connectivity between insula and sensory areas, maybe you would see increased rates of that, increased amounts of that in PTSD. So I think it's one of those questions that until we start asking the question the right way that it's answerable, we may not see it. But I think your point is really, really well put. It'd be great to hear more of it.
Thank you. Yeah, that was a great smorgasbord, as you mentioned. I was interested in the galvanic sensors. Because it does seem quite scalable, as you mentioned. I guess my question is if we are able to identify folks who do have some amount of like predisposed risk to developing PTSD while they're kind of in that more like learned fear phase, as opposed to when we usually get them in the kind of like entrenched expression of fear phase, like how do you see that impacting like our treatment of those folks?
Yeah, no, that's great. Thank you. So, our favorite model is if you right now have chest pain or arm pain, if you have chest pain, you go to the emergency department and with an EKG, with some cardiac enzymes, maybe with a CT scan, can determine if you're having an MI, early stages MI or early stages stroke, and very specific interventional approaches to potentially prevent the long-term sequelae of heart problems, right? So if we had the right predictive analytics that within a few hours of your trauma, we could say, yeah, you're at 95% likelihood of having PTSD 6 months from now. Would you like to take this treatment? What might that treatment look like? It might be one of the potential armamentarium of drugs we're starting to think about. The ideal might be could you take something that first night that would both help you sleep and diminish that threat consolidation, because that first night of sleep is so critical. One of the things we're seeing some actually interestingly unexpected data with propranolol and prazosin the first night, in which it actually looks like they may increase fear. So while those are some of our better treatments later, we're worried that we may be preferentially decreasing consolidation of hippocampal and declarative networks over amygdala networks, which again could be really bad, right? So we want to decrease emotional memory consolidation, but not declarative memory consolidation. So we have some work to do. But in terms of what that could look like, you could imagine a drug or a therapy or an biofeedback or something that you might do the first day or the first week or two after the trauma or on the battlefield to prevent the long-term sequelae. Yeah.
Thank you, Kerry, for a great talk and being a tough act to follow. You talked really nicely about the expansion of the notion of trauma from experiences on the battlefield to those experiences in disadvantaged neighborhoods. And I was wondering just what your thoughts are on the idea of complex PTSD as sort of a way of reconceptualizing borderline personality disorder.
Yeah, so I have I speak at Louis Troy Kane's annual BPD conference on this issue. So so complex PTSD, if you're not familiar with that term, is the basic idea of people generally who've had developmental trauma in addition to later lifetime traumas, having the classic PTSD symptoms, but also having much more in the interpersonal dysregulation, much more emotion dysregulation, issues with attachment and these sorts of things. And for all the world, many symptoms that we call borderline personality disorder as a trait look like complex PTSD symptoms. The borderline personality experts would argue that there's likely a significant subset of people that we diagnose with borderline personality disorder that are likely complex PTSD, but there are probably some people who are not that, and like everything else, it's probably heterogeneous. But certainly, it raises the important point from a treatment perspective that whereas our primary treatments now for BPD are things like dialectical behavior therapy, they're really emotion regulation skills, which is great, but if there's underlying trauma that we aren't targeting, we may be leaving a lot on the table in terms of the causative components of people's symptoms. So I think all I can say is, yes, I agree. I think there's definitely a trauma component that we don't understand there that we need to treat. I think it also raises a broader important question, is that we know from both human and animal studies that we're developing emotional memories from very early after birth. And if you look at like Regina Sullivan's studies and others, looking the first days after birth, we don't develop our declarative memory systems until 3 years of age or so. For the most part, nobody has memories, before about 3. So that suggests there's 3 years of life where we're developing emotional memories in terms of attachment, in terms of response, and other things, that we have no declarative tools for top-down regulation. And so I think a lot of what complex PTSD may be is where you're getting this disconnect between emotional memories and the ability to regulate them with declarative memory systems. And I don't know that we have good treatments yet for that. I think in the back. Yes, ma'am, you have the mic. Yes, ma'am.
Okay. Hi, Princess Felix, University of Michigan. Thank you for your amazing talk. It brought up a lot of questions about predisposition that I had for you. So you talked about how there are genetic markers for predisposition for risk of developing PTSD. But it was really interesting when you talked about how inhibition of CRH could reduce the aggressive response, but not really the social interactions. And you called the aggressive response, it's kind of correlated with the PTSD-induced fight response. And I want to know, there are differential responses to PTSD. So there's fight, flight, and freeze, right?
Right.
So are there individual differences, maybe even genetically, that could predispose one to these different responses? So one person might be more predisposed to the fight response, while one might be predisposed to the freeze response.
Yeah, it's a terrific question. Thank you. I don't know the answer to that. I think, as with everything, for trying to keep this to 60 slides, which I still went over, I took out a lot, and one of them was my gene by environment slide. And I think, everything in psychiatry and psychology is gene by environment. I think PTSD has the advantage of everybody who studies PTSD has been studying the environment for, as long as we've been studying PTSD, because it was, it's by definition an environmental disorder, and we're just now starting to understand more of the genetics and the biology. I think the subtypes of PTSD are also going to be, gene by environment. And so my guess is there's probably going to be some genetics that may predispose more to an externalizing versus internalizing, if you will, threat response. But almost certainly what the trauma was and how one's personality state and other things will also interact with that for whether one is more likely to be a fighter or to try to hide. And I think, a lot of that, as we understand more, is, both the dose response curves of distal threats versus proximal threats and what that interpretation is, what one's interpretations of self-agency are. There's many ways one could dive into that question, but almost certainly there will be personal differences that probably are both nature and nurture. Thanks.
Kerry?
Yes, please.
If I could just make a comment. It was a great talk, and
Thanks,
I just
sir.
want to comment on this as
Please.
well. I think the aggression piece is really interesting and novel. And, I think, further understanding how aggression plays out in relation to PTSD and anxiety in general is going to be really interesting. One comment in relation to your question: in our work with non-human primates, we have a paradigm where we can elicit defensive aggression.
Nice.
And we've studied that and find that individual differences in that are quite stable over time. And there's a whole range of individual differences. So I think that sort of plays into sort of the issues that you're talking about and perhaps predisposition for different types of responses. But I think it's going to be really interesting to get more into the aggression area, and especially the CRH tie that you
Yeah.
talked about was particularly interesting.
Yeah, thanks, Ned. I'm just starting to figure out how to talk about it, so I'll need to make sure I look up some of your papers and add those in. That's great. I think we had a couple more. Oh, maybe you first.
Yeah, I have a question. Thank you so much for your talk. And I am interested in general about social communication and how that is helpful for mental health and resilience against like developing things like PTSD. And so at the start, when you were talking about social support being a very protective factor, I'm curious why you think that is so protective, and if it's helpful with emotional regulation skills and stuff, or if maybe that's like, I don't know, if like past generations were really socially interactive, that is like genetically like helping you, or just what your thoughts are
Yeah,
generally.
it's a wonderful question, and it's a terrific question. It's also, sort of an obvious question that, interestingly, the field hasn't tried to dissect very much at a mechanistic level yet. I guess I could throw out a few things at multiple levels. I think at the interpersonal level, if your trauma were personal, interpersonal, there's, that schism of not being able to trust is so critical. So I think the more data, if we go to cognitive behavior terms, the more data you can build up in your brain that humans are trustworthy, the more probably buffered you are against generalizing that one person's trauma against me means that all people are not trustworthy. It's almost like latent inhibition towards, yes, most people are safe, and yes, there's one person that I need to remember. So another way I think about it is that the brain, I think I don't have good data for this, but my own personal hypothesis is that the amygdala is designed for one trial learning of threat and to generalize that. And the hippocampus and cortex's job is to then carve out safety everywhere else. And if you don't have good data opposing that, you're going to be left with things are not safe. I think for all the other, maybe there's generational components that would be really interesting. I think at the basic level, something that's also not that well understood, but there's some really interesting data on oxytocinergic systems in the central amygdala. And Ron Stroop had shown that oxytocin activating receptors in the central amygdala decreases fear and threat responses. And of course, unfortunately, many of us were new and were friends with Larry Young, who passed away recently. Him with Tom Insel were some of the leaders in understanding oxytocin and pair bonding and oxytocin and social support. And so my guess is that part of safety is that oxytocinergic social buffering system embedded within our brain, and that probably helps as well. But I think a lot of research needs to be done.
Cool. Thank you so
Thanks.
much.
Over
I don't know.
here.
Okay,
I was really impressed with the work that you did imaging the CRH neurons in the amygdala,
thanks.
and seeing the firing of the neurons before the behavior,
Yeah.
and also using the dreads to turn off the neurons and then seeing the change in behavior.
Yeah, thank you.
That would suggest that a CRH antagonist
Yeah.
might be really good
Yeah.
for the treatment of PTSD. Do you know if there's been any large-scale clinical trials that examine the effectiveness?
Yeah, Ned and I could probably have many beers with you over the sad history of the CRH antagonists at the pharmacological level. Basically, the CRH hypothesis is not only not new, it's one of the oldest hypotheses we have for, probably 40 plus years now. And Charlie Nemiroff, one of my mentors, did a lot of work in this area for a long time. And so long before the genetics, there was evidence that increased CRH in the spinal cord was associated with depression and PTSD, and on in all the animal studies. And yet, when we got to large scale studies, the CRH antagonists that we think cross the blood-brain barrier, that's an important asterisk. And we think we understand the pharmacokinetics of important asterisk, but at least it doesn't cause liver damage. It's got all the safety and none of the efficacy. Didn't show effects. The problem was in those studies, it was all comers. And almost certainly, again, as I've mentioned a couple times, PTSD is going to have tens, if dozens, if not hundreds of subtypes. And almost certainly there's going to be some subtypes that are responsive to the antagonist and not. And part of the problem right now with translation that we've been trying to help address with ACMP is bringing FDA together with industry, with academia to say, if at the end of the day, we've got decades of neuroscience and genetics progress, but the FDA still requires us to have heterogeneous depression as our outcome measure, heterogeneous PTSD as our outcome measure, how are we ever going to iterate these findings to targeting subtypes of disorders? So that's a side conversation. But the take-home measure is the studies were not effective. I think there's still great promise in these. My one other caveat is that the more specific we get with understanding cells, circuits, and peptides, et cetera, the harder it is for things to work. And it's kind of like the tricyclics, the dirty psychiatry drugs work better than the very specific psychiatry drugs. I think we're going to have to get to a point, for the most part in oncology and infectious disease, you don't treat with a single agent because the systems are too smart and they evolve to work around it. I think we're going to have to say, okay, if we want to target the fear off neuron, I need to hit the neurotensin receptor, I need to hit the CRH, I need to hit FOXP2, I need to hit that system in multiple ways so that the brain can't work around it for homeostasis. So I think we're going to have really a neurobiologically driven multi-drug approach with a lot of these systems. So that's just a lot of random thoughts about probably being defensive about why the CRH antagonist didn't work, but Nate can tell you more. I don't know who has a microphone. Whoever has a microphone, speak next. Oh, here. So you and then you. Thanks. Yeah, maybe front, gentleman in the front and then woman in the back.
Okay. Hi, fantastic talk. I really enjoyed that. And I was curious, you were talking about kind of a distinction between a dissociative and non-dissociative type of PTSD. And I'm particularly interested in like TMS targets. And I was wondering if you think between those different subtypes, whether or not you would want to have different targets. Like I know left DLPFC is a common target for PTSD, and just about kind of efficacy across those subtypes.
Yeah, it's a great question, and it's what people are thinking about. there's a lot of excitement with neuronavigation and some of the approaches of really doing much more specific targeting, though I think in reality, most of our clinics are still just hitting the DLPFC because it mostly works and it's hard to do the large enough size studies to pick apart the other reasons. But, our canonical models are that that classic hyperarousal PTSD is overactivation of the amygdala and deactivation of the DLPFC and the prefrontal, and dissociation is hyperactivation of precuneus, some of the sensory systems, and deactivation of the amygdala. So that model would suggest that we should not treat them the same way, because further deactivating the amygdala may simply just be sort of furthering that emotional numbing. But exactly what we would want to do, I'm not sure. We're seeing overall less brain connectivity, less functional connectivity across brain regions and dissociation, suggesting there's sort of more internalization, more default mode. So we may have to treat dissociation more likely would severe depression or something like that. But I think we're still trying to figure that out. But it's a great, I think using that as a first split, I think, some of the first, if we were to say, what are the subtypes that are probably real, it's going to be dissociative versus not, and probably childhood trauma versus not. And then from there, it's going to be biological subtyping, I think. Yeah, thanks. Oh, yeah, yes, ma'am.
Thanks for your talk, Dr. Kessler. So I guess my question is kind of similar to his question. So there's this big discussion in clinical psychology and psychiatry about how diagnostic boundaries can be blurry and then artificial, and how we should take a transdiagnostic approach to mental disorders. So I think your talk speaks to how much within group heterogeneity there might be, right, within PTSD. But I'm also curious if you can comment on, just based on your work on the neurobiology and genetics of PTSD, whether there's any crosstalk between PTSD and other disorders, which a symptom-based classification like the DSM may not get at.
Yeah, absolutely. Great question. In addition to my cough overuse metaphor, my other one is that half the genes in the genome are expressed relatively specifically in the brain. We have literally thousands of discrete disorders of the body and the organ systems, and yet roughly our neurobehavioral disorders, we have like 6, right? You have affective disorders, you have anxiety disorders, you have psychosis, substance abuse, and a couple more. So clearly there's room for these to be broken down 1,000 ways, even if you just say, okay, the number of genes associated with disease. The first level answer to your question is in the million-person GWAS. There's about 80-something% polygenic risk sharage with depression, and maybe 70-something% with schizophrenia. So the first level, what we're learning from the large-scale genetics is that psychiatric disorders are more alike genetically than they are different. So if you compare psychiatric disorders to, say, Parkinson's or Huntington's or Alzheimer's, they cluster together. Within psychiatric disorders, schizophrenia and bipolar cluster, depression, anxiety, and PTSD cluster, but you're still getting some, and there are certainly some loci that look very strong for depression and aren't there at all for PTSD, and are strong for PTSD and not there for depression, but there's only a small number of those relative to the total number. So, I think what we think will happen is a lot of the great twin and family work that has led to sort of ACE analyses and other sorts of, environment versus ancestry analysis over the years can now start to be integrated with the genetics analysis and say, okay, these are genes that are truly sort of shared pathways for general risk in psychiatric disorders. These are ones that seem relatively specific for this or that. But I think how do we cluster all of that and use machine learning and other approaches is still at the very beginning of what we're starting to see. Great, thanks. Yes, we're in the back.
Hi, thanks. I want to first say thanks for a wonderful talk. It was really a master class in working across different levels of analysis.
Thanks so much.
It's amazing. Connect to genes and circuits. And I wanted to kind of connect the previous question with Richie's initial question. And I want to ask you what you think about pain. And what we're seeing is in the UK Biobank, genetic overlap between shared risk for PTSD and multiple kinds of chronic pain in the body. That goes back to Bessel van der Kolk, I suppose.
And insulin.
Yeah, and the insulin. And it seems like though, that a lot of your analysis and focus has been on cues, reactivity to cues, and so forth. But what about the idea that also reinforces systems? These traumas often created because of physical injury and pain to the body. So maybe we're also sensitizing reinforcers that can then heighten the
the negative part of the cycle for maybe any subsequent stimulus?
No, I think it's a great question. I think there's a small but burgeoning literature on unconditioned stimulus sensitivity. So differential pain responses associated with differential PTSD. You know, Sam McLean from the Aurora study is a pain doc originally. So we're starting to integrate. We did all sorts of quantitative pain assessments in the same time we were doing all this other stuff. So we're just starting to integrate the pain data in the Aurora longitudinal study with the more classic PTSD data. And then at the animal level, again, and so in other states, and seeing you definitely, you know, as you have more unconditioned, as your shock is more intense, you get more PTSD-like symptoms, right? So I think there's all these levels of interesting intersection that we don't quite know yet. And then on the follow-up side, the reinforcer or not, you know, I think across almost all disorders, including all psychiatric disorders, including pain, avoidance is so core, right? And whether it's pain avoidance or threat avoidance, and certainly, you know, whether it's OCD or whatever, that cycle of avoidance is as much part of the functional disorder as the internal symptoms themselves. And avoidance both prevents extinction from happening and leads to dysfunction. So I think there's so many untapped levels of overlap between pain and trauma and fear that it would be really fun to talk more. Yeah, thanks. Okay.
First of all, thank you for a great talk. I know you mentioned there's a correlation between early childhood trauma and PTSD. So I was wondering if we were able to identify early childhood trauma in military members, would it be a possibility of preventing the development of PTSD if they were to experience a traumatic event?
Yes, and it's also politically extremely complicated. So Ron Kessler, who I think everybody knows of, who I have an interesting alliteration. So we're collaborating on a few things, and I'm KR and he's RK, but I'm always very honored to be called Dr. Kessler. So, and so since his work for decades now with the military and the Million Veterans Program and Army STARS, some of the earliest data was the biggest predictors of suicide, depression, and PTSD in the military was childhood trauma or prior depression or anxiety disorder symptoms prior to going into the military. We kind of know that from epidemiology. We ethically don't want to be telling people they can't serve. The Army doesn't want to decrease the number of people who might serve, and yet we know this is a risk group, right? And so I think the natural place to go would be to identify them and then work on early treatments or stress inoculation or prevention or early arousal. But I think for the most part, the military's more tried to just pretend it's not, I don't know. It's a very strong observation that I don't think the military's known what to do with yet.
Yeah, I ask you because I'm in the military, so it's you're not allowed to say all these things because you can get discharged or whatever. So we're working on a project like this in our class, and we're wondering if there's any research done or
there's a lot of research. Turning those findings into action, I think, is going to be hard. I mean, DARPA, DoD, Air Force research they're putting increasing efforts on how do we take stress pathways and known risks for stress pathways and train resilience. And so perhaps that's one way they're trying to identify it. But today, the simplest thing would be to say, if you have high risk factors, you probably shouldn't be going into combat. But
thank you.
Yeah, thanks.
Yeah, thank you so much for your talk. I was wondering if you had any thoughts about how we can use pro-neuroplastic agents like ketamine and psilocybin to facilitate fear extinction as opposed to fear reconsolidation, or if that's possible.
Yeah. Well, I think that's I don't know, given current investments, the however many billion-dollar questions. I think first being aware that this is a risk is important. You know, things like MAPS training, you know, I think, you know, there's a lot of, I was just at ADA last week and at a couple different psychedelic symposia and talking with some of the folks. While, you know, I think the biggest problem with the field right now is we don't know how to placebo control this stuff because you know you're on it. But I think the second is the therapies that they're really sure and feel really good about are these very intensive, like all day long exposure therapies. Well, we know, you know, in Australia, if you have spider phobia and want to get it fixed quickly, you go do full flooding and you spend about half a day, you know, when you start with the spider across the room, and by the end of the day, you're holding a spider. We know for years that mass training works and probably drives extinction over sensitization. But what the exact parameters of that need to be, we don't know. But I think being aware of that and really looking at that, and as per earlier, finding better quantitative markers as opposed to just self-report is going to be really critical. Maybe one more question? Is it? Yeah. Yeah, thanks.
Please hear you talk.
Oh, thank you so much.
It's more of a meta question to contextualize all that you've been talking about today. As someone who works a lot and presents a lot to patient-facing populations, which is not common in academia, there's been a huge trend in, I guess I'm calling an over-identification with PTSD and trauma and going far beyond the diagnostic boundaries, which of course there's questions and always revisions, but there's kind of a narrative now that everyone has PTSD, everyone has trauma, every single adverse experience or stressful life event is trauma based on context and self-identification. So as a resounding expert in PTSD, I'm curious on your conceptualization and what you would respond to the way people are seeing it now.
Yes, for better, for worse, trauma has become a TikTok sensation, right? So I'm going to actually know someone who I won't mention, but who's a wonderful person, an amazing therapist, and got sort of TikTok called out because they were basically saying some of the people who were sharing their own stories about dissociative identity disorder, for example, really didn't meet the clinical criteria diagnosed for that. And then they were basically publicly shamed and a huge pushback that they were being insensitive. And so it's a complex world we live in right now when everybody's an expert. All I can say is that most of our studies are done with people with very severe life-threatening trauma. And while that stress, we're in a world in which stress is pervasive, we definitely are in a world in which we've evolved this brain for basic fight or flight, fear, survival, and yet we live in a 24/7 social world that feels equally threatening to our brain, but we don't know what to do with it. That we are at a very interesting time in the history of humanity where we are feeling both the positive and the negatives of socialization in ways that we didn't evolve to. That said, we try to keep at least the research focused on things that are very clearly on the far end of trauma exposure, because I think that middle ground and certainly the mild ground is so complex that it's hard to make progress there. So I'm not on social media. I try to stay out of this because I don't know what the right answers are, but that's where I come from, that I don't know the answers to all those things. I'm sure there's a lot of people suffering. We try to focus this work on much more explicit understanding of trauma exposure.
That's more of a civil answer than I could think of, and I'll just quote you from
Thank you so much. Thanks, everyone. Really appreciate it. So we have a break. Thank you so much. Those were great questions.